Assalamu alaikum, dear students. Welcome to my new session. In this section, we will talk about the basic chemistry of protein. Before starting my lecture, I have already recorded to 05 lectures on different topics. So, first of all, you will have to review all these lectures, which is very important for each and every students those who are studying in medical colleges or in other health institutions. So they're important for all my students who are in Pakistan or they're across the globe. This lecture is specially designed for BSN students as well as Form D, BDS, and all those who are studying the health sciences and different organizations. Okay, so let's take a start from the basic chemistry of proteins. In this lecture, we will cover the basic objectives which are already you can see here on my screen. We will talk about the general chemistry of amino acids, its composition, and the amphoteric nature of protein, dipeptide structure of proteins, primary, secondary, tertiary, and quaternary structure of proteins, the function of proteins, biological importance of proteins, its solubility, composition, properties of protein, colloidal nature, and denaturation of proteins. So first of all, generally, the general chemistry of, as we know that amino acids are the monomers that make up proteins. It means that the building blocks of protein, they are amino acids. Without amino acid, the protein's existence is not possible. Each amino acid has the same fundamental structure, which consists of the carbon atom, which are known as the alpha carbon, which is bonded to the amino group or carboxylic group that is called COOH, and the hydrogen atom. According to its composition, the amino acids are the molecule which are used to build proteins. All the amino acids have a central carbon atom surrounded by hydrogen atom and carboxylic group, and amino group as well as alkyl group. So it is the R group or the side chain that differs between the 20 amino acids. Amino acids often refer to the building blocks of proteins, which are the compounds that play a critical role in your human body. So as in previous chapters, we have already talked about that, let's suppose if nucleotides are the building stones of DNA. It means without nucleotides, you are unable to design or draw the structure of DNA. And the same case with carbohydrates, which are the complex organic compounds composed of carbon, hydrogen, and oxygen. And the same case with lipids, which are the heterogeneous group of compound related to fatty acid. So it means that the fatty acid, these are the building blocks of the lipids. Come toward the amphoteric nature of the amino acids. How we can differentiate between carbohydrates, proteins, vitamins, macronutrients, micronutrients. So, protein, this is also one of the major class, which is the nitrogenous group of compounds, which is composed of multiple number of amino acids. Now, if you overview the amphoteric nature of the amino acids, so the amphoteric nature of amino acids are amphoteric compound as they contain both the acidic carboxylic group and the basic group that is called amino group. They can react with both alkalis and acid to form salts. This occurs directly. This occurs as a result of dissociation of carboxylic group to release the hydrogen ions which passes from the carboxyls to the amino group. A sweater ion is a molecule that has at least two functional groups, one having a positive charge and the other having a negative charge with an overall charge of 0. Zwitterions can form from other molecules which are called ampholytes or amphoteric compound and can act as both an acid and base. So as we know about the dipeptides, which is a short protein consisting of only two amino acid links together by one peptide bonds. While in multiple different dipeptides can be made using dehydration synthesis, which remove a molecule of water, resulting in the formation of the peptide bond. Now let's see here. This is the structural formula of different compounds, especially in proteins. You can see here this is peptide bond, and which the carbon is attached with the oxygen by double bond, and here the nitrogen is attached with hydrogen by a single bond. And this one is alkyl group, and this is hydrogen, while the nitrogen is attached to two hydrogen atoms. Now look at here, in these conditions, this bond, this is called peptide bond. As usually peptide bond is the bond which is present between the amino acid and protein molecules. So the same condition is here. So this is the overall structure of the dipeptides. Come toward the structure of protein. So protein structure is a three-dimensional arrangement of atom in an amino acid chain molecule. Proteins are the polymers, specifically polypeptides formed from the sequence of amino acids, the monomers of the polymers. A single amino acid monomers may also be called a residue indicating a repeating unit of polymer. As you can see the different structure of proteins. So this one is called primary structure of protein, and this is the backbone, while this one is the side chain, and this one is the secondary structure, in which this structure is called alpha helix structure, while the third one is tertiary, and the fourth one is quaternary structure of protein. In primary structure of proteins, the protein refers to the sequence of amino acid in the polypeptide chain. You can see here there is also polypeptide chain which is present between all these amino acid, all these protein molecules. The primary structure is held together by peptide bonds that are made during the process of protein biosynthesis. The sequence of a protein is unique to that protein and defines the structure and the function of protein. Now let's see here. This is a primary structure of protein, and this structure you can see the nitrogen is attached to the two hydrogen, while in this condition there is carbon double bond with oxygen, and this one is alkyl groups, and this is hydrogen. While the same case is here, here is nitrogen atom, and then there are two carbon, and one carbon is attached with the oxygen atom by double bond, and then this is alkyl group, and this one is hydrogen. So this whole structure is called primary structure of protein. So in another sense, this is also the primary structure in which the amino, this is called amino end, while this is called amino acid subunits. Actually, there are 20 different type of amino acids which I'm going to give special names. That is, the first one is glycine, this is proline, threonine, glycine, uh, threonine, glycine, and this is lysine, cysteine, proline, leucines, valine, and many and more. Aspartic acid, alanine, valine, arginines, glycine, prolines, and alanine. So there are different type of amino acids which are attached with one another. This is the primary structure from 1 to 5 and then 5 to 10 and this then 20 to 15 and then 20 and so on. And the secondary come toward the secondary structure of protein and secondary structure of protein. The elements typically spontaneously form as an intermediate before the protein folds into its three-dimensional tertiary structure. The secondary structure is formed defined by the pattern of hydrogen bonds between amino, hydrogen and carboxylic oxygen atoms in the peptide backbone. This is the secondary protein structure. You can see here this one is alpha helix and the second one is beta fleeted sheet is present. So I think these are the branch and you can say this is very large compound. So you can see the structure and with the alkyl group is present and the same condition, the same situation, they are also repeated here. So here is a carbon double bond with oxygen and here is nitrogen is attached. So on both sides, on one side carbon is attached and then hydrogen is attached. So this is the different bonding situation. So it need little bit practice how to draw the different structure of the protein molecules or amino acids. So you will have to practice, do some exercise on it. Okay, now another one is tertiary structure protein. So in tertiary structure protein tertiary structure is the three-dimensional shape of protein. The tertiary structure will have a single polypeptide chain big bond with one or more protein secondary structure in the protein domains. The interactions and the bond of the side chain within a particular protein determine its tertiary structure. So in this structure, you can see there is hydrogen bond which is present between this is CH2 ethyl and this structure there is hydrophobic interaction clustering of hydrophobic groups away from the water and van der Waal interactions are polypeptide backbone. This one is what hydrophobic interaction and this one is disulfide bridge. You can see the sulfur which is present. So because there are two sulfurs are present, that's why it is called disulfide bridge. And here in this case, ionic bond is formed. You know that there is occur electrostatic force of attraction between positive and negative ions. Or you can say here is occur the loss and gain of electron is also occur. So depend upon on the condition. This one is CH2, CH2. So they are attached by a single bond with one another. Okay, this is ammonia, while this is oxygen and carbon double bond, and this is CH2. Now look at here, this is the quaternary structure of proteins. The quaternary structure of protein that is polypeptide chain or this one is called beta chain and this one is alpha chain and this one is iron and heme. So this is actually the structure of hemoglobin and this one is collagen. So both are protein in nature, but these are quaternary protein. It means we can easily differentiate between the primary, secondary, tertiary and quaternary structure of protein on the basis of its structure, shape and its configuration. So there are different biological functions of proteins. Proteins are the class of macromolecules that perform a diverse range of functions for the cell. They help in metabolism by providing the structure support and acting as enzymes, carrier, or hormone. The building blocks of proteins monomer their amino acids. So the proteins are also responsible. It can help in growth and maintenance, as well as it caused by chemical reactions, act as a messenger, and it provides structure to the different compounds. It maintains the proper pH. It also maintains the fluid balance in your body, as well as it can boost up your immune health, and it can transport and stores nutrients. So we can take a simple example of hemoglobins. Hemoglobin, which is a protein that is found in your red blood cells, and they are usually responsible to carry oxygen to different tissues. Then classification of proteins on the basis of protein solubility. So solubility is the amount of proteins in a sample that dissolve into solutions. Proteins recommended as a food that use can be partly or completely soluble or completely insoluble in water. Protein solubility is the first functional property usually determined during the development and testing of new protein ingredient. Proteins are built up out of amino acids. All amino acids have a similar big bond structure but different in their side chain. The side chains have different properties. Some are hydrophobic in nature, which are not water soluble. So some of them they are not soluble in water, or they are poorly soluble. Whereas other are hydrophilic. So hydrophilic are water soluble, and the protein is then not soluble anymore. Now, what are the basic composition of proteins? The basic composition of proteins, there are 20 different amino acids which make up proteins. Each amino acid consists of central carbon, and the central carbon is bonded to an amino group, a carboxylic group, and a hydrogen atom, and an alkyl group. I'll come towards some biological functions of proteins, which is important for the growth and maintenance, as well as it's caused by chemical reactions. It is a messenger, provides structure, maintain the proper pH, fluid balance, and immune health transport, which we have already discussed in our previous lecture. Come toward the certain properties of proteins. So proteins, uh, regarding its color and taste. So proteins, they are usually colorless and they are tasteless. Proteins, they have shape and size. So on the basis of shape and size, the proteins range in shape from simple crystalloids, spherical structure to long fibrillary structure, molecular weight. They have a proper molecular weight, as well as they are colloidal in nature. Denaturation phenomena has occurred, while some of them are amphoteric in nature, and while some of them there is ion binding capacity and solubility. According to the colloidal nature of proteins, so proteins are the complex macromolecules with dynamic conformations. They are charged like colloids, but unlike colloids, charge is heterogeneously distributed on their surfaces. There is amino acid size, structure, denaturing, strength, stored energy, and biological process. All these are the colloidal nature of the protein. Now you can see here, this is very good examples. The example of stable colloids, and with the particles they are equally distributed in a proper sequence. So there is no fluctuations among these colloids. And this is the example of an unstable colloids where the particles, so they are so aggregated and stuck with one another, and the particles they are settled down. So you can see sedimentation of these particles occur. So this is the classical example of unstable colloid which doesn't maintain its equilibriums. It's the arrangement or the consequence or the sequence their distribution of these molecules, they cannot maintain its equilibrium. So there is sedimentation occur, and the particles they are aggregated and or they are sandwiched in one another, you can say. So this is a very good example. You can see it practically. Denaturation of protein, it is another phenomena which is occurred in the proteins. Denaturation is a process in which the proteins or nucleic acid lose the quaternary structure, tertiary structure, and secondary, which is present in their native state by application of some things. So this is all these things are very easy to understand, but it need little bit practice to do it. Okay, so uh, epiprotein loses its shape, so it ceases to perform that function. The process that causes a protein to lose its shape is known as denaturation. Denaturation means when the protein molecule causes to lose its shapes. Denaturation is usually caused by the external stress on proteins. Primary, secondary, and tertiary structure we have already. Okay, so I was talking about the denaturation, which is usually caused by external stress on the proteins such as solvents and organic salts, exposure to an acid or base, and by heat. So by heating, proteins can directly cause denaturation. What are the factors that affecting the denaturation? So the first one is temperature, then pH, then salinity, polarity of a solvent. These are some other factors that influence the shape of protein. So if any one or combination of these factors varies from the normal condition, so the shape and the function of the protein will change. And this change in the shape is called denatures. So definitely if the temperature of the protein can be enhanced or increases, so there will be occur the denaturation, so there will be occur conformations, there will be occur changes in the arrangement, especially of the quaternary, secondary, and tertiary structure of protein. Now you can see this is a good example, denaturation of protein when the protein lost its biological activity. And this one is normal protein, and this one is denatured proteins. So after denaturation, the shape, the property, the physicochemical property, the arrangement of the atoms, they are completely disturbed. Again, when the denatured protein it become normal, so its shape become like this. Okay, there are some other examples and classification of proteins based on conformation base. There are some protein, for example, fibrous and soluble in water. That is alpha keratin, beta keratins, and collagen. The globular solubles, which are solubles in water, that is myoglobin, hemoglobin, lysozyme, ribonuclease, and chymotrypsin, cytochrome C, lactate dehydrogenase, and peptidecene. On the basis of composition, one is simple protein, then conjugated, and then derived proteins. So simple protein is albumin, globulin, glutellines, prolamines, protamines, histones, and many more. While another one is the conjugated one is nucleoproteins, lipoprotein, phosphoprotein, metalloprotein, glycoprotein, flavoprotein, hemoprotein, and so it means that the nucleoproteins, these are the conjugated protein in which the nucleic acid is joined together with the protein molecule. While in case of lipoprotein, the lipid is attached with the protein. While in case of phosphoprotein, the phosphorus is attached to the protein. And metalloprotein, metals atoms, for example, if iron is attached with the protein, then it is said to be metalloprotein. And glycoproteins or carbohydrate is attached to the protein, while in many and more, so all these are the example are based on the nature of, so the acidic protein is the proteins which are present in your blood, that is acidic protein, while the proteins which are basic in nature, that is histone, and histone they are also found, these are basic at pHs, basic. So it was this our discussion was about the proteins, amino acids, and I think this lecture is very important for all medical students. So thank you so much for watching my lectures. So if you have any question or if you need this content, so I'm always available for your services. So kindly email me. So inshallah I will immediately give you a reply to all my students. Okay, thank you so much for watching my lecture.